The first thing we'll discuss in this talk is the concept of a biosignature. So a biosignature is defined as some material, chemical, or characteristic of a planet that is believed to have its origins solely in organic processes. So this means some gas or chemical that we might find on a planet that can only be created by the chemical reactions that occur in living organisms. So biosignatures on Earth might include methane gas, which is produced in agricultural settings as well as by humans, and carbon dioxide. But the presence of these molecules doesn't necessarily guarantee the presence of life, because there are other processes that are not organic that also create these gases. So this picture shows a couple of hypothetical planets where different chemicals may be detected. And while some of them may appear to be biosignatures, the presence of others basically make that hypothesis false. So for an example, the central plot here, we have O2 molecules, which is oxygen as we breathe, O3 molecules, which is ozone present in our atmosphere, and H2O, which of course is water. So these three molecules are all chemicals that would be present on a potentially life-bearing planet. However, this O4 molecule, which is circled here, would actually be evidence of a separate chemical reaction that creates water, oxygen, and ozone, and is not organic. So in that case, finding the O4 molecule would actually show that life is likely not what is causing these chemicals to be present on this planet. Similarly, the lack of detection of other molecules, which are shown down here in the bottom, would also indicate that these chemicals could be produced from an inorganic source. The next type of biosignature that I'll talk about are called surface biosignatures. So surface biosignatures include changes in the light that we observe from a given planet that is due to something organic on the surface absorbing parts of that light. So an obvious example on Earth would be the way that the vegetation that grows here reflects green light. So there are obviously the sunlight comes with many colors, but only the green light is reflected in leaves, and in some other leaves, some other wavelengths are also reflected, but largely vegetation reflects green light. So what this means is that an image of Earth would show a much brighter intensity of green light as compared to other light. So this can indicate the existence of things like chlorophyll or similar organic molecules that are actively absorbing energy from the host star. The final biosignature I'll talk about are so-called temporal biosignatures. So temporal more or less just means biosignatures that change in time. So one such biosignature on Earth that is a temporal biosignature is the changing measurements of carbon dioxide in our atmosphere. So as the seasons change and plants go through their normal cycle of growing in the spring and then decaying in the fall and winter, the amount of carbon dioxide they take in changes. And so that ultimately represents itself as a changing measured carbon dioxide abundances in our atmosphere. Similarly, as the seasons change, the so-called albedo or reflectiveness of our planet changes. So snow reflects light in a different way than vegetation. So different seasons make Earth show up differently. So these are all evidence, measurable evidence that we could detect on other planets as well. So unfortunately, because Earth is our only, or maybe I should say fortunately, Earth is our only life source that we know of. So we need to use Earth as a test subject for many of our presence of life metrics, as well as studying how life develops on Earth. So to begin that discussion, I wanted to go back to the study of abiogenesis. So this is the study of the origins of organic life coming from non-organic or inorganic compounds. So, of course, this is something that humans have wondered about for centuries, if not more than that. And honestly, there's still lots of speculation and questions around the original sources of life on Earth. One pretty popular opinion is the so-called primordial soup hypothesis, which more or less said that in very early Earth times, a collection of various atoms and elements that had accumulated on Earth's surface over the millennia of time leading up to that point, slowly, because of the environment where they were living in early Earth, or they were not living in early Earth, they grouped up because of the electric forces between them and eventually formed spontaneously early chemicals and early building blocks of amino acids and other organic molecules. So this hypothesis was actually tested later in the 50s with the Miller-Urey experiment. So this apparatus on the left side of the screen is the so-called Miller-Urey apparatus. So this experiment essentially tried to reproduce the conditions on early Earth, with many of the chemicals we expect were present in Earth's atmosphere at that time and on Earth's surface, as well as the very violent environment from regular lightning and radiation from the sun. So all these things existed on early Earth, and this experiment wanted to see whether or not it was possible that inorganic materials could spontaneously group together into organic materials, or at least the building blocks of those organic materials. And this experiment was actually fairly successful. So after about a week of allowing this apparatus to do its thing, several different amino acids were formed in the trap in the bottom of the apparatus. So this was the first time that we had ever shown that early proto-organic materials could be developed in an otherwise inorganic environment. So another way that we have used Earth as a test bed for early life and the progression of life is just to look at how life has developed in the wide variety of environments present on Earth. So while we live generally in fairly comfortable environments, life still manages to find a way in the most awful, hot, acidic, oxygen-poor environments on Earth. So this includes inside volcanoes, at the bottom of the ocean, in trenches and incredibly hot and high-pressure vents at the bottom of the ocean, and deep within the frigid ice in the Antarctic. So all these places, while they are incredibly uninhabitable for us, are still habitable for other organisms. And in fact, many of those organisms thrive in those environments. So I've included a couple pictures here of some other specific examples. So this specific on the right here, this specific organism is called a deep sea xenophyophore, which is a sort of fungus-like creature that lives at the bottom of the ocean. Similarly, the tardigrade, a fairly popular organism in science fiction and biology community, is a very hardy, small organism that is found on Earth and has managed to survive flooding and living in space and being frozen. So it has adapted to live in these harsh circumstances, which means that we would expect life to be successful at living in other environments as well, though this still doesn't really tell us much about the generation of life so much as the propagation of life. We also can use some more astronomy-related measurements to study Earth's habitability and the effects of life on Earth. So one such case was the Galileo spacecraft, which its primary objective was to observe Jupiter and its moons, which it did successfully for about 8 years. But on the way to Jupiter, the spacecraft took several images of Earth, and those images, some of those images are actually shown here. So this bottom image is an image of the Australian coast, and this red along the coast is actually increased methane concentration. So methane is generated as I mentioned before, as a product of agriculture and just human life. So increased amounts in an environment on Earth where we also have oxygen shows that this gas is being created regularly on the planet. We can say that because methane and oxygen will react with one another. So the presence of both suggests, especially in this abundance, suggests that something is generating that methane. On the right here, we have another image. This one is of the Nile River at different times in the year, where you can see changes in both the liquid water on the surface, as well as the albedo and methane production from plant life and other organisms living near the Nile. So these are the kinds of things that we might expect to actually be able to see on other planets that host life. Finally, the last thing I'll talk about, which I thought was very interesting when I read about it, is the concept of using Earth's Earthshine. So Earthshine here is actually the light that has reflected off of Earth's surface and then reflected again off of the moon's surface back to us. So we can observe the moon and measure subtle differences in how our light is reflected by the moon's surface. And those subtle differences actually tell us about the changes in Earth's albedo, which, as I mentioned before, is the reflectiveness of our planet. So as a year goes by, our planet is reflective by different amounts. In some months where we have lots of snow and ice, the planet becomes more reflective than in those times where we have more vegetation on the planet. So all of these things allow us to use Earth as the one test subject for life and how life affects the universe around it in ways that we might be able to measure on other planets. So to continue on that thread into other planets, our next video will be covering some of the other planets in our solar system, as well as other planets that we've learned about in the last few years.